Let's start. Okay, today we have Peter, who's been with us for 3 months. We've been working with Melo and Amparo, counting a lot of agarrobas. And today he's going to present his work. He's been he performed for his master thesis just before going back to Switzerland.
Yeah, no, first Austria, then Switzerland. Okay, thank you very much, Tim, for the friendly introduction. I really like the fact that I got the opportunity to present here my master thesis, also because, like, yeah, it's basically presenting in front of people I know, presenting in front of my family. That's really cool. And so it happens that the weekend is around the corner, and the sooner we start, the sooner it's over and the weekend starts. So I would suggest we get right to it. Like Tim said, I will present my master thesis. The title of my master thesis is Bee-Plant Interaction and Dependence of Honeybee Density and Landscape Structure and Agroecosystems. The thesis was supervised by Thomas Frank and Sophie Kratschmer, and we will start with a short introduction. Since all bees forage on nectar and pollen, and therefore there is potential for increased interspecific competition. Interspecific competition refers to competition between different species, and they compete for resources, which is in this case a limited resources, because in this case the resources are, as said, nectar and pollen. And every landscape provides every day a specific amount of nectar and pollen, which is available for the bees to forage on. And 1 unit of nectar foraged by one bee is not available for another bee. And that's why they compete for the same resources. And wild bees have, in comparison with honeybees, competitive disadvantages because of their way of life. The main difference is that honeybees, they live in huge hives together, like many thousands individuals, and most of the wild bee species are solitary, and by that I mean that they live on their own. There's no hive, no colony, only one individual in charge of everything. And yeah, when we think about that. Like ourselves, if we want to do something, if we do it together, it's easier and goes faster. And if you do it on your own, most of the time it takes longer and not so fast. And that's why, and that's where the competitive disadvantages come from. The main competitive disadvantages are, for example, they have shorter activity ranges, so the distances they can overcome from their nest to the I don't know, to the flowers they forage on. They are, in comparison with honeybees, way shorter. And also since they're alone, there's no communication between individuals. For honeybees, for example, they have a quite complex way of communication in between them. For example, scout bees leave the hive, look for floral resources, they find them, they come back and they do their fancy dance, and then the others know where to go. And so this makes foraging for honeybees way more efficient. Wild bees, they just go out, they find something, they don't find something, just like that. So it's way less efficient. And wild bees do not build up food stocks. So honeybees, they build up food stocks in order to bridge times where not enough floral resources can be found within the landscapes. We also like their food stock. It's called honey. But wild bees, they do not produce honey at all. And also the beekeeper helps honeybees to overcome such times without sufficient nectar and pollen supply by just moving them to another place where they can find enough plants, floral resources, or by just feeding them. But at the end, the contribution to pollination of wild plants and crops is complementary. So we need both of them. We need wild bees and honeybees. And this dynamic raises questions about what are environmentally sustainable honeybee densities. And with my thesis, I tried to address this dynamic through two research questions, one of which is how do various landscape parameters alter the level of specialization and the level of shared resources within the plant networks in follows in agroecosystems. And the other research question is how do many landscapes parameters affect wild bee species richness and abundance in agroecosystems. Now we've come to the geographical scope. In total, I had 19 study areas located in Eastern Austria, and they were sampled from April until the end of August 2021. Here in figure 3, you can see on the auto photo the exact location of the study areas, and down there at the bottom left, where Austria is located in Europe. And one thing that might be of interest for you, Eastern Austria, so this point more or less here, is not located within the Alps. It's in the Austrian lowlands. And it's Austria's wild bee biodiversity hotspot. This is due to the climate because it's quite warm there. No, actually, it's very hot during the summer there, and during the winter, the temperatures don't get like super low like we know it from within the Alps or in the mountains. So this is also why and the precipitation there is. The mean annual precipitation there is around 500 millimeters, so also quite dry. And that's why you can compare this continental climate a little bit with the Mediterranean, which is why you can also find some Mediterranean species there. But you have to be very lucky to find them because the populations there are very, very tiny, let's just say that. And those species I was talking about here in the Mediterranean, you can really find them in high abundances. So that was also one idea why I came here in order to take a look at them. But anyway, and also like here on the auto photo, you can see that there are some weird circles drawn around my study areas, and this has to do with the experimental setup I came up with on each follow. And in order to explain this a little bit, how the experimental setup looks like, we will take a closer look at this with this schematic representation. In every follow, we place the center of a study area. Around that, we created the study area with a circle with a radius of 500 meters. And we chose 500 meters because the activity range of most wild bee species is 500 meters. And then within the study area, we placed the study sites. So the places we sampled, where the sampling took place, which was in my case, 1 transect and 5 plots. So all the sampling we did took place within the study area. And also around the center of the study area, we created the catchment area with a radius of 1,300 meters. We chose this distance because this is the distance around apiaries where native plant pollinator interactions are significantly altered, more or less, like up until 1,100 meters, not 1,300. It took a little bit more. And also within the catchment area, the landscape parameters were a point of our interest. Okay, moving on. Now I will talk a little bit about the sampling method, starting with the transect method. The transect was 100-meter long and was sampled by walking the whole length over a period of 20 minutes. And all bees that were on the left or the right of the transect were catched. And this was done in order to assess the bee-plant interactions on follows. Now I'll talk briefly about the plots, the sampling methods of the plots. In total, 5 1 square meter plots got placed at locations where many of the selected flowering wild plants were blooming. And each study plant plot was sampled by observing it for 10 minutes. And later on, for the statistical analysis, all of those plots, they got pooled per study area and per selected studied wild plant. And now that I've mentioned so many times selected wild plants, I have to talk a little bit about the selection process and which plants we selected. We had certain criteria for the selection. One very important criteria was that the plant was in general attractive for bees, and also that it was a plant which could be frequently encountered in our Eastern Austrian agroecosystems. And also the flowering time was very important because we don't have a wild plant which flowers throughout the whole vegetation period. So in order to sample each month a plant, we had to find one flowering wild plant per month, and we came up with Prunus spinosa in April, Anthemis austriaca in May, Medicago sativa in June, and Carduus acanthoides in July. And the plots were sampled in order to assess the flower visitation of bees on the selected plants. I've also mentioned landscape structures and landscape parameters. And in order to assess them within the catchment area, field mapping of mass flowering crops was done, like buckwheat, oilseed rape, and sunflower. And also the field mapping of apiaries was done within the catchment area in order to come up later on with the honeybee density around our study area. And also the amount of semi-natural habitats got determined through the Korean land cover data and the Invecus data. Yeah, I feel like that's enough said about the landscape structures for now. And we've already arrived at the results. We will start with the bee plant networks on my photos. In total, we found 605 individuals belonging to 28 different bee species, and 280 out of those 605 individuals were honeybees, and 325 were wild bees. And here on the left in figure 8, we can see the bee plant network for May, and on the right side we can see the bee plant network for July. Taking the interactions, the complexity of interactions into account, we can see that the main network is way more complex in comparison with the July network, because here in the main network we can't find any plant species, for example, which is of central importance for most of the or all of the bee species present within the network. Whereas here in the July network, we can find plant species which play a central role. For example, here Daucus carota played a central role for bee species with short tongues, and also Carduus acanthoides. It looks like it played in general a central role, since around 50% of all the bee species found in the network were also found on foraging on Carduus acanthoides. Yeah, and also honeybees were quite frequently encountered on Carduus acanthoides. And now here in table 1, we see that the level of shared resources peaked in July, although on a very, very low level. Just to explain it a little bit, this niche overlap index, it varies between 0 and 1. 0 would mean no niche overlap, 1 would mean the perfect niche overlap. And since we have the peak of it is 0.2, there's not so much overlapping going on when it comes to resource use. So we can't really start talking about or thinking about interspecific competition, which has negative negative effects on wild bee species or populations. But we figured that since also in other studies they found in midsummer an increase in interspecific competition, we figured that in a low way, I found this in my master thesis too. So in July, if for example in Austrian agroecosystems interspecific competition with negative effects could appear, it might probably occur in July. That was what I thought about this. And also while interpreting this those bee plant networks, we have to keep in mind that while I was sampling those networks on my fallows, we had mass flowering crops going on within the landscape. In May, for example, oilseed rape was flowering, and in July, sunflower were flowering. And that's why here in table 1, we can see the percentage of area covered by those mass flowering crops. And it is interesting that even though like almost double the area was covered by some sunflower in comparison with oilseed rape, that in July still many honeybees wanted to forage on wild plants on fallows, because normally the normal behavior of honeybees would be that they forage in the most efficient kind of way, and they prefer mass flowering crops over the other plants. But in this case, it was not like that. Why could that be? For example, I read in another study that honeybees, they can determine which pollen is the best pollen when it comes to nutrition. And so I was thinking maybe Carduus acanthoides, when it comes to pollen quality, provided superior pollen, more nutritious pollen in comparison with sunflower, for example. Okay, and now we will move on to the results of the bee flower visits on selected wild plants. In total, 861 individuals were found belonging to 83 bee species. Out of those, 233 were honeybees and 628 were wild bees. And as we can see here, the distribution of wild bee and honeybee individuals on the wild plants was rather different. And in the next slide, we come back to this distribution, but in a different kind of form. And so I will give you a short explanation of how that could be or why it was like that in a few seconds. Here the effect of the selected wild plant on wild bee species richness and abundance showed also quite quite different outcomes, let's just say that. And in comparison with Prunus spinosa, this effect was significant and positive for Carduus acanthoides, and for Medicago sativa, it was significant but negative. And in order to explain this a little bit, we might find some possible explanations when we take a look at flower morphology and the flowering time. With flowering morphology, I mean how does the flower look, the shape of it, the form, the dimensions, stuff like that. Because for bee species to forage in an efficient way on Medicago sativa, they would need a very long tongue. And for Carduus acanthoides, in order to forage efficient on it as a bee species, you would neither need a long tongue nor a short tongue. It's quite easy to get access to the pollen and the nectar. And so in general, since not all bees have especially long tongues, this would mean that potential more species could forage on Carduus acanthoides in general, in comparison with Medicago sativa. And also, I mentioned the flowering time. Medicago sativa was flowering in June, Carduus acanthoides in July. And this means that, yeah, the rhythm of the bee species, the annual rhythm of the bee species is that in July, the amount of bee species active at the same time peaks, at least in Austria. I feel like here in July, it's way too hot and there's not so much activity. And so since we have this peak in July, there are even more species active, which could possibly forage on Carduus acanthoides. And this could, yeah, from my point of view, explain those differences found. Yeah, no effect of honeybee density on wild bee species richness and wild bee abundance was found. But in order to discuss this a little bit, I have to question the quality of my honeybee density data. I was doing the field mapping within my catchment areas, and a circle with a radius of 1,300 meters is quite big. And so it took me like almost a month in order to finish it. And also probably maybe I haven't found all of those apiaries located there. And when we think about that, apiaries, they get moved throughout the vegetation period to I don't know, from mass flowering crop to another mass flowering crop, by also sampling the honeybee density only once per year. It is questionable that my honeybee density data really showed what was going on in the real world, for example. Yeah. And yes, the effect of mass flowering crops on wild bee species and wild bee abundance was significant and negative. Since wild bees also get attracted to some extent to mass flowering crops, it seems logical that if you have a fallow enclosed by mass flowering crops, that the wild bee abundance on your fallow gets decreased, and also the species richness. From my point of view, this just seems quite logical. Now here we have the interacting effect of honeybee density and mass flowering crops on wild bee species richness and wild bee abundance. And it showed that a negative effect of honeybee density could get amplified as mass flowering crops and honeybee density increases. And since adjacent mass flowering crops could also increase the honeybee abundance in fallows, it might be that in order to forage in regions where you don't encounter so many honeybees, also wild bees tend to forage on mass flowering crops. So that might be one explanation. Oh, it looks like, I don't know, that was a fast presentation. Okay, already arrived at the conclusions. We concluded that wild plants provide an important food resource for wild bees and honeybees in Eastern Austrian agroecosystems, especially after the flourishing of mass flowering crops. And increasing the amount of floral resources secures the sufficient nectar and pollen supply throughout the whole vegetation period. And it probably would also reduce the potential interspecific competition. And we figured out that in spring, Prunus spinosa was very important for the species active in spring. And in summer, Carduus acanthoides was important for the bee species active in July. And for example, when we think about of doing something for the bee species active in spring, we could think of promoting Prunus spinosa. For sure, we can't concentrate us only in Prunus spinosa, but the thing is, Prunus spinosa is a bush, it grows in hedges, and since due to the intensification of our agroecosystems over the last few decades, we lost quite a few hedges, and bringing back hedges and also Prunus spinosa, which would come with this, seems like a good idea. And in summer, Carduus acanthoides, it has shown that in July it would be most likely that if interspecific competition with negative effects appears, it would appear probably in July. So if you find yourself in such situation, you could think of I don't know, promoting Carduus acanthoides or any other plant which is bee attractive and could grow there in this specific month, probably. But those are just ideas. We could later on discuss about that. We would not recommend to promote Medicago sativa due to its flower morphology, because I already read about studies where they showed proof that planting Medicago sativa as a pollinator promoting measure, you only increase the abundances of quite frequently encountered wild bee species anyway, because it's only attractive for if you have a long tongue. So the sad thing with Medicago sativa is that in Austria, this is a subsidized agro-environmental scheme, and it is listed as you do something for your soil and for the pollinators. And if you want to do something for your soil, plant it, perfect. But if you also have in your mind, I do something good for the pollinators, you better plant something else. That would be my opinion on that. But yeah, in general, further research is needed, and hopefully this thesis could help implementing future research projects, as the main function of my master thesis was producing a manual for the implementation of future projects, and I learned quite a lot of what to do, what not to do. And now I've already arrived at the end of my presentation. I would like to thank you all for your attention, and my deepest gratitude to all the people who have helped me through this. And I would end it now by showing you a few impressions of my summer full of field work in Eastern Austria agro-ecosystems. Thank you. Okay.